Hello friends, today we are going to solve 970013 October-November 2023. So first question, which statement about light microscopy is correct? The options are, as the resolution increases, the magnification increases. There is no relation that magnification increases with resolution. So magnification is something which makes the object bigger, and resolution is something how clearly we see the object. uh, Statement B, as the magnification increases, image always becomes clearer. No, when magnification increases, image will become blur unless and until the resolution is very clear. Option C says that the resolution will decrease as colored light of increasing wavelength is used. Yes, so resolution and increasing wavelength of colored light are inversely proportional. So as the wavelength of colored light increases, the resolution is going to decrease, and magnification and resolution are the terms that relate to the same factor. No, as I said earlier, magnification and resolution are completely different. So magnification is how big the object is, and resolution is how clearly the object is seen. So the answer is C. Question 2. The diagram shows a stage micrometer scale viewed through an eyepiece containing a graticule. The small divisions of the stage micrometer scale are 0.1 meter, and here we should know to identify which is stage micrometer and which is ocular meter. Okay, so this one, and the question they are asked for, what is the actual length of nucleus in the plant cell? So before identifying all these things, we should calculate and calibrate the stage micrometer and the stage micrometer with the ocular meter. So the top line which is shown there is the stage micrometer, and the bottom scale is the eyepiece graticule, and they have clearly mentioned that the small division of stage micrometer is 0.1 millimeter. So the distance between stage micrometer division is 0.1 meter, and from the diagram we can easily able to identify that 40 division in eyepiece graticule is equal to 0.1 millimeter. So to calculate or to find the value for one division in eyepiece graticule, we should simply calculate using the formula by 0.1 divided by 40 gives us 0.0025 millimeter, which is also be told as 2.5 micrometer. The nucleus corresponds to 10 division, and it is seen in the from the diagram. It is very well clear that the nucleus occupy 10 divisions in the eyepiece graticule. So to find the actual length of the nucleus in the plant cell, we simply want to multiply this 10 into 2.5, which is equal to 25 micrometer. So the answer is option B. Question 3. What is the correct order of the order for the involvement of the cell structures in the synthesis and secretion of an enzyme? So they are asking us to find the correct order for the synthesis and secretion of an enzyme. So the organelles that are given are cell surface membrane, Golgi body, nucleus, rough endoplasmic reticulum. So initially they are asking for secretion and synthesis of enzyme. So initially what happens, so nucleus first involves in the process of transcription, that is mRNA synthesis. From mRNA, after synthesizing mRNA, it is sent through the rough endoplasmic reticulum. From rough endoplasmic reticulum, proteins will be sent to Golgi bodies for further processing, and finally to the cell surface membrane. So the order will be 3 4 2 1. So it is nucleus, rough endoplasmic reticulum, Golgi body, and cell surface membrane. Question 4. Centrioles, cilia, microtubules, and microvilli are structures found in eukaryotic cells. Which statement about these cellular structures are correct? So centrioles composed of microtubules and not cilia and microvilli. So first statement is wrong, and centrioles play important role in mitosis and semi-conservative DNA replication. Centrioles play a role in mitosis but not in DNA replication. And cilia and microvilli both increases the surface area for absorption. Microvilli increases the surface area for the absorption of nutrients, but cilia does not increase the surface area for absorption. Cilia is just there for the movement of to and fro movement, which sweep away the mucus. Then microtubules are made of protein and form the spindle in mitosis. So option D is the correct one. Question 5. Which description of plasmodesmata is correct? So they are channels through plant cell walls lined by the cell surface membrane. Plasmodesmata are channels in cell wall lined by cell surface membrane. Yes. Then they are channels through plant cell walls and that are formed from proteins. They are not formed from proteins. Then they are channels required for the movement of water through apoplast pathway. Apoplast pathway, water move by space in the cell wall without entering the cell. Then they can be lignified and form pits in the walls of the xylem vessel. So plasmodesmata can never become lignified. That's why in the pit region there are cellulose cell wall will be there still in xylem, which helps in the movement of water from one xylem vessel to the other xylem vessel. So the option is A. So the correct answer A. They are the channels through plant cell walls lined by the cell surface membrane. Question 6. Which cell organelle does not have nucleic acid? So it's a straight question. Which cell organelle does not have a nucleic acid? So chloroplast, yes, chloroplast and mitochondria, they have their own DNA. And we have from the rest of the two options, Golgi body and ribosomes, we have to identify which two organelle doesn't have their nucleic acid. So as we see, ribosomes also have their rRNA. So rRNA is also a nucleic acid. So the only option is Golgi body, which doesn't have its nucleic acid. Question 7. Which process occur in eukaryotes and prokaryotes? So the list of process what they have given are hydrolysis, mitosis, transcription, translation. So mitosis does not take place in prokaryotes. Instead, binary fission will happen in prokaryotes. So all the bacteria and the prokaryotic organism will divide by the process of binary fission and not by mitosis. So mitosis, for the process of mitosis to take place, nucleus should be there. Then the remaining process, hydrolysis, transcription and translation, takes place in both. So the answer, leaving off 2, all the other 3 options are right. So the answer is C, 1, 3 and 4. Question 8. Which bonds are present in all viruses? Covalent, ester, phosphodiester. So not all viruses have ester bonds. Viruses that have lipids have ester bonds, but covalent and phosphodiester bonds are seen in all viruses. So the answer is C, 1 and 3. Question 9. The diagram shows the structure of a monomer. So which molecules contain this monomer? So first we need to identify the monomer given. So the given monomer is alpha glucose, and the given options are glucose, sucrose and amylopectin. So this is present in all sucrose, glycogen and amylopectin. So the answer is B. Question 10. A triglyceride consists of a glycerol and 3 different fatty acids, linoleic acid, oleic acid and the palmitic acid. So the diagram shows one possible arrangement of fatty acids L, O and P in the molecule. So they have given first arrangement, and what is the total number of different arrangements of fatty acids in the triglycerides is being asked. So the total number of arrangements, here we cannot go by the permutation method, because as however we arrange, the structural orientation will be same for the all the other things. So LOP is the order given, and the second possible order can be OLP, and the third possible order can be LPO. Fourth order is not possible. As the molecule rotate, it will be the same. So the answer is 3. 11th one. Which statement about phospholipid molecule is correct? They contain 1 saturated fatty acid and 2 unsaturated fatty acid. they contain 3 phosphodiester bonds, they contain hydrophobic regions and hydrophilic regions, they contain a hydrophobic phosphate group that is soluble in water. So the first statement, if it is not specific to have one saturated and two unsaturated, it can be in any way. So the fatty acid can either be one or two unsaturated, or even more unsaturated fatty acids can be present, which will be called as polyunsaturated fatty acids. So they contain 3 phosphodiester bonds. Phosphodiester bonds are seen in nucleic acid. uh, Phospholipids have both hydrophobic and hydrophilic regions. Yes, so phospholipids, as the cell membrane, they have this hydrophobic and hydrophilic regions, which are water loving and water hating regions. These statements is the C1, they contain hydrophobic region and hydrophilic region. So the answer is C. Question 12. Hydrogen bonding explains many of the properties of water, including the high latent heat of vaporization and high specific heat capacity. For which processes in plants is hydrogen bonding in water important on hot sunny day? So the question clearly states that it is when it is important for, why it is important for hot sunny day. So preventing denaturation of enzymes in leaf, yes, it can come. Reducing water loss by evaporation, okay, it can also be considered. Allowing leaves to cool down quickly at night. The question clearly states why it is important on sunny day, and we are not worried about how it is going to be in night. So statement 3 can be rejected. Holding the column of water in xylem vessels together, so this can also come. So water prevent the denaturation of enzyme, then reduce the heat of reduce the water loss by evaporation. No relation between leaves cooling at night and hot sunny day, so as I stated, so statement 3 will not be coming. Then fourth one, hydrogen bonds helps water to run in a column in xylem vessel together. So the correct answer is B, excluding the statement 3. 1, 2, 4 are correct. Question 13. Which feature of cellulose molecules contribute to the function of plant cell walls? So they are asking the feature of cellulose molecule that contributes to the function of plant cell wall. So adjacent cellulose molecules linked by glycosidic bond. Adjacent beta glucose. So cellulose doesn't have cellulose molecule itself, it is made of beta glucose. So beta glucose are the only monomers which are linked together by glycosidic bonds. Branched chains of beta glucose molecules are linked by hydrogen bonds. Beta glucose is not at all branched, they are just straight link, or they just form the straight chain, so they will never be a branched chain. Option B is also wrong. Molecules of alpha glucose are linked by 1,6 glycosidic bond. Here the cellulose molecules are formed only by beta glucose, so the alpha glucose does not even come. So option C is completely wrong. So option D, the only left out answer is option D. Unbranched chains of beta glucose are linked by hydrogen bonds. Yes, so the beta glucose which forms the straight chain, or the straight chain, it will be interlinked by the hydrogen bonds. So the answer is D. Question 14. Which statement describe some enzyme actions? So the three statements given are enzymes holding reacting molecule so that their reactive groups are close together. In an enzyme catalyzed reaction, more molecules have sufficient energy to react than without the enzyme. Reaction catalyzed by enzymes takes place at lower temperature than they would without the enzyme. So according to statement 1, enzyme hold the reactive groups together, that is the enzyme substrate complex, for the reaction to take place. Yes, statement 1 is right. Statement 2, in the presence of enzyme molecules, they have sufficient energy when compared without the enzyme. And statement 3, enzyme catalyzed reaction lowers the activation energy. So when a reaction is taking place without the presence of enzyme, the activation energy will be high. When the same reaction is taking place with the presence of enzyme, the activation energy will be lowered. So all three statements are right. So answer will be A, 1, 2 and 3. Question 15. Influenza virus has an enzyme called neuraminidase, which breaks down glycoproteins in the surface membrane of the cell that the virus will infect. So the glycoprotein binds to the active site of neuraminidase by induced fit. Which statement about induced fit hypothesis of enzyme action is correct? So here we have to look only for the main important portion of this question is which statement about the induced fit hypothesis of enzyme action is correct. So from the given three statement we should compare which will go well with the induced fit hypothesis of enzyme action. So statement 1, the active site must have the same shape as the substrate for them to bind together. Active site and substrate should have complementary shape and not the same shape. Then this enzyme is less likely to be affected by non-competitive inhibitors than an enzyme working by the lock and key mechanism. So enzymes affected by non-competitive inhibitors, like they have not given any proper information or enough information about that for us to identify. So statement 2 will not come. And the substrate is converted to product by specific R groups in the active site, just like the lock and key mechanism. Yes, both induced fit and lock and key mechanism, substrate will be converted to product by specific R group at active site. So the correct answer is D, 3 only. So statement 3 is the only right answer in this. Question 16. So the graph compares the effect of an temperature on the activity of protease enzyme peptide when in solution, that is free, and when immobilized in alginate bead. So here they are comparing both free enzyme and immobilized enzyme. So which statement about the effect of immobilization of peptide is correct? So it alters the shape of peptide's active site at higher temperature. Immobilizing the enzyme does not changes the active site actually. So at high temperature, if it is going to change the active site, the reaction cannot take place. As the active site changes, substrate cannot go and bind with it. It decreases the activity of peptide at higher temperature. At higher temperature, the activity is still high for the immobilized peptide enzyme, so which shows that it is not going to decrease the activity. Then C option, it increases the stability of peptide at higher temperature. Immobilizing the enzyme increases the stability of enzyme as it can work best even in the higher temperature. As you can see, the free peptide enzyme is about to reach 0 at 80 degrees Celsius, but still the immobilized peptide enzyme is at great height. Then immobilizing, like it reduces the number of collision of peptide with the substrate. So immobilizing enzyme reduces collision between the substrate. No data given about this, so there is no data claiming that it has reduced in the collision, all those things. So from the given question, the option which goes well with well for this is C. So it going to increase the stability of peptide at higher temperature as the activity is high, still high for the immobilized peptide at 80 degrees Celsius. Question 17. The diagram shows how adrenaline activates processes inside a cell. So adrenal molecule is shown, and the question they are asked is which row is correct. So type of receptor molecule and result of ligand binding to the receptor. So the receptor molecule, the options given are phospholipid, phospholipid and protein for the type of receptor. The receptor molecule can never be phospholipids. So A and C both can be eliminated, and from the options B and D we can see that once the adrenaline attached to the receptor, it undergoes conformational change that activates G protein, which starts the enzyme cascade. So it is going to be option B, which is the receptor molecule is made of protein, and the result of ligand binding to the receptor is the receptor changes the shape to activate the G protein. Question 18. The statements are comparisons of endocytosis and exocytosis, and the question asked is how many statements are correct. So 4 statements are given. Both are mechanisms that involves vesicles or vacuoles and transport of material across the cell surface membrane. Yes, so both are going to endocytosis and exocytosis. They involve vesicles or vacuoles, and both mechanisms occur to allow bulk transport across the cell surface membrane. Yes, by bulk transport, by the process of bulk transport only, the vesicles, the substances will be moved out of the cell surface membrane or moved in through the cell surface membrane. Endocytosis involves taking materials into the cell, whereas exocytosis involves release of materials from the cell. Yes, endocytosis is taking in, exocytosis is releasing out. Some of the cell surface membrane is lost when endocytosis occurs, and there is an increase in cell surface membrane when exocytosis occurs. Yes, so when endocytosis occurring, the cell surface, like the cell surface membrane will be lost as the substances will be moving out. Along with the cell surface membrane, some few portions will be pinched off. Then again, in case of exocytosis, the cell surface membrane will be increased as the new substance, the vesicles will come and join, then it releases inside the cell. So that time the cell surface membrane will get increased. So all the given four statements are correct for both endocytosis and exocytosis. So the answer is D. Question 19. A student was asked to calculate the surface area as to volume ratio for an agar cube with a side length of 5.5 millimeter. Which surface area as to volume ratio is correct? So, to calculate the surface area as to volume ratio, cube is equal to 6a square by a cube. So, by using this formula, we can easily identify this answer for this question. So 6 into 5.5 the whole square divided by 5.5 the whole cube. So which gives like 181.5 divided by 166.375, which is 1.09 by 1, which is close to 1.1 is to 1. The closest value is 1.1 is to 1. Even simply this can be calculated by cancelling a square and a cube. So this gives 6 by 5.5, which is equal to 1.09 by 1, and again the closest value is 1.1 is to 1. So the answer is D. Question 20. Which statement about the behavior of chromosome during telophase is correct? So we need to check the behavior of chromosome during telophase. So chromosome attached to spindle fibers at the equator. So chromosome generally attached to spindle fiber only at the end of prophase or during the anaphase, and chromosome starts to uncoil inside the newly formed nucleus. So during telophase, chromosome separates and uncoil inside the newly formed nucleus. Statement C says, option C says, chromosomes move towards the opposite poles of the cell. Chromosome move towards the opposite poles only during anaphase. Then option D, chromosome condense into compact structures in the cytoplasm. Chromosomes condense into compact structures at the start of mitosis. So the correct answer is B, chromosomes start to uncoil inside the newly formed nucleus. Question 21. The transmission electron micrograph shows a cell in a stage of mitotic cell cycle. So here they are asking which statement explain why it is difficult to identify the stage of mitotic cell cycle shown. So chromosomes have supercoiled and are visible, but centrioles are not visible. Chromosomes are not supercoiled in this given image, and anaphase may be continuing or telophase may be starting. From the electron micrograph, it is clear that cell is dividing and still chromosomes are moving apart. So the cell must be in late anaphase or early telophase. Option C says it is unclear whether the electron micrograph shows two cells in metaphase. The electron micrograph shows two cells but not in metaphase. Some people may consider interphase have started. Only if the cells are separated, interphase will start. So the answer is anaphase may be continuing or telophase may be starting. Option B is the right answer. Question 22. During lagging strand replication. Short fragments of DNA are produced, which is the Okazaki fragments. The fragments are joined together by DNA ligase, and which row correctly describes the structure of these short fragments of DNA. So the conditions they have given are they have the base sequence that are complementary to the newly synthesized leading DNA strand, and they consist of one polynucleotide strand, and they contain only carbon, hydrogen, oxygen and nitrogen. So if we see all the three conditions, we need to say which statement goes well with uh, the Okazaki fragment or the short fragments that are being produced. So lagging strand will have base that are complementary to the synthesizing leading strand. Yes, definitely they are going to have complementary strand to the newly synthesizing leading strand, that is the newly formed or newly producing Okazaki fragments will have bases that are complementary to the newly synthesizing leading strand, and they consist of one polynucleotide strand. Lagging strand have polynucleotide strand. Yes, they will be having polynucleotide strand, and they contains only carbon, hydrogen, oxygen and nitrogen. They contain carbon, hydrogen, oxygen, nitrogen, and also phosphate will also be there. So option D is the right answer. So they have base sequence that are complementary to the newly synthesized leading strand is right, consists of one polynucleotide strand is right, and contains only carbon, hydrogen, oxygen and nitrogen is wrong, as it also contains phosphate. So the answer is D. 23. The genome of the bacterium Escherichia coli has been altered to enable it to code for an amino acid that is not found in nature. All the ATC DNA stop triplets on the strand of DNA that is transcribed have been substituted to ATT. The ATC triplet can then be inserted to code for the new amino acid. A new tRNA can then be constructed to carry the new amino acid. So the main or the major information that we want from this question is that the ATC triplet is inserted again to form a new amino acid, and a new tRNA can then be constructed to carry the new amino acid, and they are asking, the question asked here is what is the anticodon for this new tRNA. So to have a tRNA, to have tRNA anticodon will bind with the mRNA. To have mRNA will be formed from the DNA template. So ATC is the inserted DNA triplet code. So the mRNA strand for ATC is UAG, and tRNA anticodon for UAG will be AUC. We can also find this in a simple way, like DNA and tRNA anticodon have the same basis, but the only thing where they differ is the T in DNA strand will be replaced by U in the RNA. Since RNA doesn't have thymine, it only have uracil. So all T in the DNA strand will be replaced by U in an RNA strand. So we can easily find that the answer is ATC will give AUC. So option B, AUC is the right answer. Question 24. 40% of the bases in a section of non-transcribed strand of DNA are purine molecules. What will be the percentage, what will be the total percentage of cytosine and uracil bases in the primary transcript, that is transcribed from the other strand of DNA? So here purine 40% is in the non-transcribed strand, and they are asking the total percentage of cytosine and uracil bases. So total percentage of cytosine and uracil is being asked from the transcribed strand. Cytosine and uracil are the pyrimidine. So purine always pairs with the pyrimidine. So since the non-transcribed strand is having 40%, the transcribed strand should have remaining 60%. So the answer is D, 60%. 25. Question 25. Which statement about xylem vessel elements is correct? A. Hollow vessels enable the constant movement of water up and down a plant. So xylem helps in the movement of water only in upward direction, not in the downward direction. Then option B. Pit enables the movement of water into adjacent xylem. Pits are the region where lignin is not deposited, so water can move to adjacent xylem vessels. Option C, vessels contain numerous mitochondria to generate ATP for active transport. Xylem is dead cell, so it doesn't have mitochondria. Option D, vessels contain perforated cross walls called sieve plates. Xylem vessels are hollow, so they don't have perforated end plates. Only phloem have the perforated end plates. So the option is B. Pits enable the movement of water into adjacent xylem vessel. Question 26. So ammonium ions NH4 plus can enter the xylem of plant roots by two pathways. In the apoplast pathway, ammonium ions move through cell walls until the pathway is blocked by the Casparian strip. Ammonium ions then enter the cytoplasm of the root cell. In symplast pathway, ammonium ions move through the cytoplasm of the root cells. Scientists measured the uptake of ammonium ions into xylem of Arabidopsis thaliana. The SGN3 mutant of Arabidopsis thaliana does not have Casparian strip. So in SGN3 mutant, ammonium ions can enter the xylem without entering the cytoplasm of root cells. So since the mutant variety of Arabidopsis thaliana doesn't have the Casparian strip, the water can go either by apoplast or symplast. Even without entering the cytoplasm also, the water can enter into the xylem. So the question here asked is, they have given the bar chart results which is showing the plant variety and uptake of NH4 plus, that is the ammonium ion, into xylem. So here they have non-mutant and mutant variety, but they have compared this in two conditions, one in low concentration of ammonium ion and the other one is in high concentration of ammonium ion. So which conclusion is correct, they are asking. So option A, fewer ammonium ion enter xylem when they have to move through the cytoplasm of root cells. More ammonium ions enter the xylem at low soil concentration of ammonium ions. The Casparian strip acts as a barrier to reduce the movement of ammonium ions into the xylem. The loss of Casparian strip has a little effect on the movement of ammonium ions into the xylem. So in both the cases, more ammonium ions entering through the cytoplasm. So that is clearly seen. Then more in low concentration of ammonium ion, less ammonium ion enters the xylem from soil. The bar is low for the low concentration of ammonium ions. Casparian strip acting as a barrier to reduce ammonium ion movement. So there is not much data given about it. And option D, loss of Casparian strip have little effect, as we can see in both low and high concentration of ammonium ions. The mutant variety is close to the non-mutant. So option D is the right answer. Question 27. Which row correctly shows processes required for the movement of water from root hair cell to atmosphere? So they have given three processes: cohesion, diffusion, evaporation. Cohesion allows water molecules to stick together during movement. Diffusion, water enter the plant through diffusion by osmosis. Evaporation, water molecules evaporate and water vapor is filled in the airspace before leaving to the atmosphere. So option A is the right answer. Question 28. Which process can be carried out by a mature red blood cell? So the answer, so they are asking for process carried out by a mature red blood cells. Active transport. Mature red blood cell lack nucleus, so they cannot do cell division. Then white blood cells, phagocytes will do phagocytosis, not the RBC. So option C is wrong. Then protein synthesis requires nucleus for mRNA production. Mature RBC doesn't have nucleus. So the correct answer is active transport. Question 29. The photomicrograph shows three white blood cells labeled X, Y and Z. Which row correctly identify these cells? So the white blood cells are labeled X, Y and Z. cell X is the lymphocyte. So it is very clear that is more than half of the cell is covered by the nucleus, so it is lymphocyte. Then cell Y is the neutrophil because it has the lobed nucleus for the process of phagocytosis. Then cell Z is the monocyte, which is the largest white blood cells. So the option is B. Cell X is lymphocyte, cell Y is neutrophil, cell Z is monocyte. 30. When a blood vessel is damaged, it can result in problems in the body. Which row identifies the blood vessel that could have been damaged? Blood is not being distributed to organs that need it. Blood is not returning to the heart and is collecting in organs. Blood surges mean that relatively constant blood pressure is not maintained. So when arteries go near to the organs, they become more muscular. Blood returning from organs to heart will be carried by veins. Then blood pressure will be maintained by the elastic arteries that are closer to heart. So the answer is D. So the blood is not being distributed to organs that need it, so the arteries which are near to the organs will be more muscular in nature. Then blood will be collected by veins that are returning to the heart. Then the blood surges that relatively constant blood pressure is not maintained, so blood pressure will be maintained at the elastic arteries which are closer to the heart. Question 31. The diagram shows how tissue fluid is formed. They are asking like what is the net pressure causing tissue fluid to flow out of the capillary at the arterial end? So near the arterial end, what is the net pressure causing the tissue fluid to flow out? So we need to find the net pressure causing the tissue fluid to flow out at the arterial end, which is on the left side. So difference in the blood pressure inside and outside is 4.3 minus 1.1, which is equal to 3.2 kPa. Then difference in water potential inside and outside is minus 3.3 minus 1.1, which is minus 2 kPa. So difference between the values of blood pressure and water potential, 3.2 minus 2 is 1.2 kPa. So the answer is C, 1.2 kPa. Question 32. What happens when carbon dioxide is transported by red blood cells? Hydrogen carbonate ions move into the red blood cells and chloride ions move into the plasma. So hydrogen carbonate ions are produced in the red blood cells. They are not going to be move into the red blood cells. So option A is wrong. Option B, chloride ions move into the red blood cells and bind to hemoglobin. So chloride ions move into the red blood cells, but they are not going to bind with hemoglobin. Option C, chloride ions move into the red blood cells and hydrogen ions move into the plasma. Chloride ions move into red blood cells, but hydrogen ions do not move into plasma. They are going to bind with hemoglobin. Then option D, chloride ions move into the red blood cells and hydrogen carbonate ions move into the plasma. Yes, so chloride ions move into the red blood cells and also hydrogen ions move into the plasma. So this forms the chloride shift. So option D is the right answer. Question 33. What is typically found in trachea, bronchi, bronchioles in the human gas exchange system? So cartilage, ciliated epithelium, squamous epithelium, squamous endothelium. Cartilage is present in trachea and bronchi but not in bronchioles. Ciliated epithelium cells are present in all three, trachea, bronchi and bronchioles. Squamous epithelium is present in alveoli, and squamous endothelium, no cells are found like that. So the correct option is B, ciliated epithelium. Question 34. The photomicrograph shows the section through a part of the human gas exchange system. Which structure is shown? So they have given four options. So first, to check with this, we have ciliated epithelium cells over there. Then we have cartilage. So the structure, they are asking for the structure shown. Presence of ciliated epithelium and cartilage confirms the structure is not alveolus and bronchioles. So since cartilage is thin and also muscular layer is seen, the structure is not trachea. So alveolus, bronchiole and trachea has been eliminated. So the trachea will have thick cartilage. So the answer is C, bronchus. Question 35. Which factors are required for the efficient diffusion of oxygen and carbon dioxide in the human gas exchange system? So they are given three statements. Clean and warm air entering the lungs. So clean and warm air has no relevant function for the efficient diffusion. Then maximize the area of exchange surface. So maximum area of exchange surface and minimum distance between alveoli. So and blood helps in efficient exchange. So both statement 2 and 3 are right. So the answer is D. Question 36. Which statement correctly identify why it is difficult to eliminate TB? So humans have limited access to clean water. Limited access to clean water. TB is not a waterborne disease. So statement 1 is wrong. Humans can be infected, but pathogen can be inactive. Yes, humans infected by TB can be asymptomatic, but still they carry the pathogen. A number of drug resistant strains of pathogen have evolved. So bacteria causing TB become resistant to antibiotics can happen. So the option D again, 2 and 3 is the right answer. Question 37. Bacteria can become resistant to antibiotics. What can help reduce the development of antibiotic resistance in bacteria? So again they are given three statements. Use specific antibiotics instead of wide spectrum. Use antibiotics to treat viral infection. Develop new antibiotics. So specific antibiotics can be used to treat instead of using many antibiotics at a time. Then viral infection cannot be treated with antibiotics. So statement 2 is wrong. And development of new antibiotics can also help in reducing the antibiotic resistance in bacteria. So option 1 and 3 is right. So option C is the right answer. Question 38. The diagram shows one way of testing the effect of an antibiotic on bacteria, and they have given a diagram which shows the petri dish with disc of filter paper soaked in antibiotic and diameter of zone measured every day for 5 days. So here they have the main information given here is the zones of less than 13 millimeter show the presence of resistant bacteria. So the type of bacteria which shows zone less than 13 millimeter are resistant to or resistant bacteria. So which statement can be supported by this data? So after going through the data we can come to a conclusion that bacteria which show zone less than 13 millimeter are resistant. So all type of bacteria become resistant to antibiotic over time. Yes, they can, but no data provided. So option A, all type of bacteria become resistant to antibiotics over time. They can become resistant to antibiotic over the time, but data in this table is not given to that. Then option B, only types 2, 3 and 4 of the bacteria show resistant to the antibiotic. According to the data, bacteria type 2, 3 and 4 shows zone less than 13 millimeter, so they are resistant. Then option C, the antibiotic can be used to treat types 1 and 3 only. Antibiotic can be used only for treating bacteria 1 and 3. No, it can be used to treat for all bacteria. Then option D, type 5 of the bacteria can never become resistant to antibiotic. Type 5 of the bacteria can never become to resistant. No, it might become resistant over time. It takes some time, or through the evolution it can become resistant. So according to the given data, the correct option here is B. Only types 2, 3 and 4 of the bacteria shows resistant to the antibiotic as they have zones less than 13 millimeter. Question 39. Some responses made by cells of the immune system to a pathogen are listed. So mitosis, recognize a pathogen, produces memory cells, secretes enzyme. Which responses are correct for phagocytes? So the question here is which are the response are the correct for the phagocytes. So phagocytes will engulf pathogen and digest them. So they do not go for mitosis. So first they should recognize the pathogen and they need to secrete enzymes to digest the pathogen. So mitosis and memory cells are responses shown by lymphocytes. So the correct answer is D, 2 and 4 only. Question 40. Some vaccines do not contain antigens. The vaccine contain a molecule of mRNA. Cells in the immune system use the mRNA molecule to make protein antigen. The statement describes the stages of how mRNA vaccines work when they enter a cell of the immune system. So the statements given are B lymphocytes and T lymphocytes with complementary receptors bind to the protein. Lymphocytes differentiate into memory cells that give long-lasting immunity. Ribosomes translate the mRNA molecule to make a protein. The cell displays the protein on its cell surface membrane. And the question here is, what is the correct order of the stages of how mRNA vaccine works? So the correct order, we need to arrange all the four statements in the correct order. So first, ribosome translates mRNA to protein. Proteins will be on the cell surface membrane. B and T lymphocytes bind to the protein. Then finally memory cells will be produced for long lasting immunity. So the answer is C, 3 4 1 2. Thank you for watching. Please do subscribe, like and comment. Give your feedback in the comment section. Thank you.